Hello again, Biology 300 students. Mr. Parker here, and again we are looking at the classic genetics, and we're on Screencast session number 4. This is on incomplete dominance, codominance, and sex-linked traits. As you can tell, one of the big main areas that we're going to talk about are the different types of blood typing. But first, let's go ahead and take a look at our learning targets. As you can see here, you should also have reference pages are chapter 8.3 and 8.4 in your book, and then also some of the slides will be in your packet on page 133 through 136, and also 139 through 140 in your packet. I'd also have out your screencast diagram sheet or kind of graphic organizer sheet, so you can have some note-taking area, and we're going to run through some of the different topics here. So, first one is incomplete dominance, and what you can tell by the picture here is that when we talked about complete dominance, we always had a dominant trait and a recessive trait. So when we're talking about phenotypes, we always had two. But now when we get into the idea of incomplete dominance, there's a blending that's occurring. So kind of the heterozygote form is going to have its own phenotype. So we're going to moving from just having one phenotype or two phenotypes to actually having three phenotypes in incomplete dominance. So the basic definition here is dominant gene does not fully express itself to mask the effects of the recessive gene. And as you can see here, again, it does not fully express itself. A pattern of inheritance in which the phenotype of a heterozygous individual is intermediate between those of the parents, and the genotypic ratio, which is a key point here, the genotypic ratio is now equal to the phenotypic ratio. So as you can tell here, we have our initial cross that's occurring between the two parents, the purebreds here, and then in F1 generation, we are going to see all the pink, or the combination of all the hybrids, and then when you cross all the hybrids, you're going to get what we have here is the 1:1 ratio, and that ratio is the same now for genotypic and phenotypic ratio. So genotypically we have our 1:1, and then phenotypes we're looking at, we have red versus pink versus whites. So phenotypically we have the ratios there that you can see, and so the incomplete dominance, again, the main thing is the dominant traits no longer mask the recessive traits. When you have a combination of the two, you're going to get a blending. You go from having only two phenotypes to actually having three phenotypes now in this case. So again, just kind of looking at the three different types. You can see we have the dominant here and the recessive, and then we had the combination of the two. All right, moving into the second topic here is what we call codominance. And in codominance, we are looking at here that two or more alleles are fully dominant. When present together, they are both expressed, and we have ABO blood groups exhibit codominance. So when we're talking about it, your type A and your type B are dominant traits. When they're linked together, that's where you get your codominance. The picture you see on the right here is an actual electron microscope picture of red blood cells. And another picture here, you can see this is actually a capillary that's torn open, and the blood is basically escaping through here. All right, so looking at the different types of blood, all right, we have type A blood, and so if you're type A blood, you have an A antigen. Remember, antigen is basically your body, it's a protein, or what we refer to as a surface protein, so on the surface of your blood cells, and this is what your body recognizes as being as its own. And then your body produces what we call antibodies, or what we refer to as antibody B. And what that is going to do is if you ever get any antigen B in your body, or blood type B in your body would fight it off because it recognizes it as being foreign. And if you're a type A blood person, you can get blood from a group A or a type O person. Type O person is referred to as a universal donor, and we'll talk about that here in a few minutes about why they're a universal donor. But you can get blood from an A or an O person. So on this chart on the bottom, here's type A, And when you look at type A, here we have our antigens, and here we have our antibodies. So if you're type A, you have your A antigens and you have antibodies B. And if you're type A, genotypically you are either an AA or an AO. Okay, and you're still considered to be a type A blood person. All right, moving on. Now on to type B. Type B person has antibody. Okay, so they have antibody B. Sorry, antigen B, and they have antibodies A. So it's the exact opposite of what type A blood was. And if you are a type B person, you can get blood from a type B, a group B, or a type B person, and also or a type O. So now we're on the second column going down here, and you can see the person is either going to, if they're type B, they have the genotype of BB or BO, okay? And they have the antigens B, the surface proteins, and then they have the antibodies A that will fight off type A blood if it comes within the body. The next one is we have looking at is your type AB, and your type AB is a combination of the two. So this is where your codominance comes in, and you have antigens A and B in your blood, but you do not produce any antibodies. Okay, so you don't have antibodies A or B, and you're also known as the universal receiver because you can get blood from anyone. You can get it from type A, type B, type O, or type A and B. It's because your body does not produce these antibodies, okay, that are present. So genotypically, you are an AB person. You have antigens A and B, but no antibodies, and that's why this main thing here, with no antibodies, you can get blood from anyone. Okay, now type O person. Okay, the type O person, you can see here, okay, type O person, they have neither antigen A or B, so they have no antigens. They're referred to as the universal donor. And the reason that is because if you do not have any antigens, a type A person or a type B person or a type AB person, the blood that you give them will just be recognized as their own because you don't have any antigens within it, any foreign bodies. But they do have antibodies A and B that are produced, okay, which means that a group O person can only get blood from a group O person. So you kind of think of it, a type O person has a kind of the short end of the stick because they can give blood to anyone, but they can only get blood from a type O person. And again, the reasoning for that is because they don't have any antigens here, they can give blood to anyone, and those people's body will recognize as being as their own. But however, they produce antibodies A and B. So if they got an A in or a B in or an A and B in, then their body will fight that off with the antibodies and basically have the blood clot up. And the genotypically for a type O blood, you are OO when you look at the genotypes. All right. So we have here just a summary again. Here we have our antigens. And again, these are our antibodies. For your type A, you can see up top, you can see the different genotypes for each one. Good little summary sheet. You have this within your packet. And it kind of tells you what antigens you have and what antibodies you have and what basically why you can get blood from certain people. This slide here shows you kind of the blood distribution throughout the United States. Okay, and the second one I have on here is the distribution of blood types throughout the world in a few different areas that you can kind of see how the blood types are distributed. Okay. Again, a little summary sheets. Here you can see, again, if you're a type A person, these are your genotypes. You're either AA or AO, type B, BB, or BO, AB, and then you have type O. And again, the reason you can't get blood from a type B person if you're an A is because you produce antibodies, okay, B, and you have those antigens in there that will recognize that as being foreign and help fight off the foreign substance. All right, what we're going to take a quick look at here Okay, I'm going to jump out to the movie here for us so we can give you a little bit more idea of what the blood types are. And I would like to, it's a 6-minute video clip, so it'll take us a few minutes to go through. And it's going to show you, why some blood types are beneficial.
His pulse is dropping. I think we're losing him. He's lost a lot of blood. He's going into shock. Get me 10 units of AB blood. Well, actually, no, you better make that type O. Well, no, maybe we should go with the B. Well, actually, maybe, which one? We're losing him. I don't know, pick one.
Sir, are you all right?
Huh? Oh, hi. Yeah, sorry. I'm here to donate some blood.
Oh, all right. Just have a seat over there and the nurse will be right with you.
Okay, thanks. I've come here to this clinic to donate some blood, but there's something I'm curious about. Have you ever wondered why they use all those letters to describe our blood? I mean, what makes A blood different from O or B? And why is it so important that doctors know what kind of blood we have? Well, it's taken centuries for us to unlock the secrets of our blood, and we've learned some hard lessons along the way. For instance, we once believed that illnesses were caused by bad blood. So to make people well again, we would drain the offending blood from their bodies. As you've probably guessed, this procedure wasn't very successful, and for a couple of good reasons. First, here's how much blood is flowing through the average adult body, about 5 liters. Now, if you were to lose a liter or more of your blood, you'd start to suffer from a condition known as shock. When you lose a lot of blood, your body can become starved for oxygen. If that blood isn't quickly replaced, you can fall into a coma and die. And that's what happened to a lot of those early patients who had their blood drained. They died. Now, in one respect, those early blood-draining doctors were correct. A lot of infectious diseases are carried by our blood. But what they didn't know is that our blood also carries the cells we need to fight those diseases. Our blood can be divided into 2 main components, red cells and plasma. It's the red cells that give our blood its color, but their main function is to carry oxygen to our tissues. Now, if you remove all the red cells from your blood, you're left with a yellow fluid. That's the plasma. And it's here in the plasma that you find the cells that help you fight infections. Now, the discovery that blood was required to fight disease gave the early doctors a new idea. Why not just transfer some blood from a healthy person into the body of a sick person? Well, sometimes this procedure actually helped, but in most cases the result was, well, fatal. What they didn't realize at the time is that not all blood is created equal. You see, a little further along in history, we discovered 2 special proteins on the surface of our red blood cells. They're called antigens, and we labeled them A and B. Then we discovered that some people's red cells have only the A antigen. Some have only the B antigen. Some have both A and B, and some have neither antigen. Put it all together and you have the 4 basic blood groups: A, B, AB, and O. I know what you're saying. So what? Big deal. Well, it's not a big deal. That is, until you try and mix some of those blood types together in someone's body. To show you the problem, let's go back to the clinic. Now, I happen to have type A blood. So let's take a sample of my blood and divide it into 2. To the first sample, we'll add a solution containing plasma from type A blood, the same blood type as mine. To the other sample, we'll add a solution containing plasma from type B blood. Now we'll mix everything together, and there's your problem. The first sample looks normal, but in the second sample, the red cells have all clumped together into a sticky mess. So what's happening? Well, floating around in the plasma of everyone's blood are structures called antibodies. Now, the job of the antibody is to patrol your blood looking for foreign invaders. So suppose I have type A blood, but I get injected with type B blood. When my antibodies see these B antigens, they think this cell is an intruder. So they latch onto it and sound the alarm. And as the intruding red cells and the antibodies collide, they stick together and form a clump. These clumps disrupt the flow of blood to your cells, and without emergency treatment, you can die. Today we prevent this problem by cross-matching a sample of the donor's blood with a sample of the patient's blood. This test ensures that the blood is compatible and that there will be no clumping. However, sometimes in an emergency, we don't have time to cross-match the blood before it has to be transfused. In this case, we have a backup plan. We always transfuse type O blood. Why type O? Well, if you remember, type O blood has neither the A or the B antigen on its surface, which means no matter what type of blood you have, the antibodies in your plasma won't think this is an intruder, and your cells won't clump together.
Alright, so welcome back, Biology 300 students. I hope that that video helped stress some more of the importance of blood types and why certain blood types can go with other blood types, and why, if you mix like type A and type B, why you will get the blood clots occurring and the molecules will kind of come together, which would not be a good thing. The last topic we're going to move into real quick here is what we call sex inheritance, or sex-linked traits. These traits are all linked off the sex chromosome, and we've talked about this before, where males are XY and females are XX. And you can see that here on the screen. The first picture there is a little electron microscope image of an XY, and here you can see an XX. And typically, it's a 50-50 shot. If you're going to be male or female, that's the probabilities because of the meiosis that occurs and the gametogenesis and separation of the X and Y from the mom and from the dad. But looking at the sex traits and how that all works out, okay, you can see here that genes linked to the sex chromosome is what we call sex-linked inheritance. And what we're talking about is up here you can see that you have the A linked off the X, you have a dominant A and the recessive A, and the recessive would be whatever disorder we're looking at, or whatever trait we're looking at, okay? And then you can see for a male, you're only going to have one of those because this trait is linked off the X chromosome. These are usually found on the X chromosome, but not on the Y, okay? And female heterozygotes are referred to as a carrier. So if you're a female and you're a heterozygote, you're a carrier. So up here, this female, she's referred to as a carrier because she does not express the recessive traits, but she does have one of the traits that could be passed on to her offspring. So during the reproduction and during meiosis, during the gametogenesis, when the egg and sperm, the gametes are separating, you can see here you have the sperm that are being separated and the eggs are being separated here, the alleles. And when you combine those, in this particular case, you can see here's a female, she's all homozygous dominant, and a female here, she's referred to again as a carrier because she has one of the large and one of the recessive traits. The male here is normal, okay, and then down here, this male would have the recessive traits because of the lowercase a that was there. And typically, that the sex-linked traits are more probable in males because, as you can see here, males only need one of the recessive traits, whereas females, she would actually have to have both of the recessive traits in order to see that. What we have here is over here is color blindness, and this is a trait that is more prevalent in males. And you might be able to see the numbers, you might not, depending on if you're colorblind or not. Okay, so we'll be doing some problems in class, and the main thing you need to make sure when you're doing these Punnett squares is that your A, okay, stays with the X as you move them into the Punnett square, and it helps keep track of the trait that's being passed along. So, wrapping this screencast up, this is screencast session number 4 for the genetics unit, dealing with incomplete dominance, codominance, blood types, and sex-linked traits. And from here, we'll move into doing some problems in class, and then eventually moving into pedigrees and kind of taking a look into that and wrapping up the unit.